Welcome. This is a series of videos that I am producing for our lab's website called iBRAINs.org that describes the research we are involved in since about 5 years ago. We are a team of researchers interested in mapping out neuropsychological and neuroanatomical changes associated with substance abuse. So our main focus is substance abuse, although we have also explored the relationship between substance abuse and personality disorders specifically, and also we've explored areas related to dementia. So let me start out with the substance abuse area of our research, and let me start out with one of our earlier studies looking at opiates. As you all know, the opiate epidemic has become huge in the United States, so it's very important to look scientifically at what happens to the brains of patients who take opiates, whether they're synthetic opioids, general opioids, or specific opiates like heroin. We are interested in seeing if there is recovery from the drug after stopping the drug. We're also interested in seeing if it makes sense to look at potential relationships between the actual changes in the physiology and anatomy of the brain, changes in white matter, gray matter in the brain in relation to function. And we've done all of this meta-analytically. So our first study looked at white matter abnormalities in long-term heroin users. And let me introduce you to the team that you're going to hear many of these names over and over again. Scott Wollman is the person who took on the project around opiates, and Scott is now focused on his dissertation. He is going to be defending a three-part dissertation on the question of opiates, and we've published two parts. We intend to publish the last part within the next couple of months. Obviously, I myself am leading the project. We also have people who have been integral to our lab. These are in general order because you're going to see them contribute in different ways to different projects. Mark Stern, Matt Hall, Jocelyn Rompergren, Chrissy Kimmel, Adlin Perez, and several other people in future studies. With that being said, let's jump into what goes on with heroin. As you might know, heroin is associated with white matter damage. Research has shown us that at the primary level, and the reasons for that potential damage is obviously related to the degradation in the myelin sheath, but also actual alterations of genes that affect myelin in people who abuse heroin specifically. There's also evidence for mitochondrial dysfunction that results in degradation in the myelin, and neuronal apoptosis, neuronal death, that then result in degradation, obviously, in the death of the axons and then breakdown of the myelin. Most commonly, what we see is we see a reduction in something called fractional anisotropy. Fractional anisotropy is a measure of the integrity of the white matter using diffusion tensor imaging. It's a type of magnetic resonance imaging tool that allows us to measure how well the white matter is allowing a different fluid to go in one direction or to diffuse in an isotropic way. So whether something is isotropic, meaning diffusing in all direction, that indicates that that part of the white matter is not doing its job correctly. It's allowing too much of the fluid to distribute in all directions. When you actually have a intact white matter, you expect higher fractional anisotropy, and to repeat, anisotropy is opposite of isotropy, which means the fluid is actually moving in one direction, and therefore higher anisotropy is actually better anisotropy. Reduction is worse. And what we see in heroin is a reduction in the left cingulate, in the right frontal lobe, and in the bilateral frontal sub-gyral regions. That's what our primary studies show us. However, there is some contradiction in primary studies, so we see sometimes reductions in the right middle frontal area, the left parietal, the corpus callosum, and these are not often replicated. So we were interested in seeing what's the most robust findings from the limited literature on white matter in heroin use. Potential reasons for the heterogeneity is the sample size, the duration of use, and the length of abstinence. So we wanted to study these as potential moderators for the effect sizes we see in the primary studies. The technique we used is intense. We pride ourselves for doing detailed and independent searches. We also put a lot of effort in looking for published and gray literature, meaning literature that hasn't been published, but people have done the research and maybe just put it in a file drawer because it was either small, maybe not accepted for publication. So we wanted to get all the information that's out there. We did our searches on multiple databases. We had 2 searchers do the searches, and then beyond that we also contacted people who we know work on the topic. We've looked at conference proceedings and found names of individuals, contacted them for data. So we have exhausted all ways of approaching getting the maximum number of articles and research on the topic. And we used a technique, a modern technique, to assess meta-analytically the neuroimaging results that come from diffusion tensor imaging. We mainly focused on morphometric imaging. We avoided a region of interest because that doesn't cover the whole brain, and we also avoided TBSS. It's a technique for assessing or for processing white matter dysfunction. However, there were limited articles on that, and we couldn't include them in our morphometry studies. Effect size sign differential mapping has quite a few positives, and one of them is that other techniques tend to put positive and negative findings, whether it's higher or lower activation, higher or lower white matter in a specific voxel. It tends to average these. Instead, with sign differential mapping, the actual maps give us a sign, allowing us to treat negative results separately from positive results. Another advantage of using sign differential mapping over other techniques is that it actually does use effect sizes, unlike other techniques that just look at activation, non-activation. So this actually calculates the effect sizes, which then allows us to combine the reported peak coordinates with different statistical parametric maps. And in a way that mimics traditional meta-analysis much closer than other techniques. We used meta-analysis, we used meta-regression and subgroup analysis in our study, and we specifically were very interested in the question, does the duration of use make a difference, and does the length of abstinence make a difference? Because we were fascinated by the question of how much recovery is there after somebody stops using heroin. And you will notice we focused on heroin in the first study, because we discovered that the literature really was mainly about heroin, and that's typically the case in in this literature that focuses on heroin. We also wanted to avoid potential confounds of using, for example, uh, legally prescribed opiates, synthetic opiates like oxycodone. And the reason is these are usually prescribed to individuals already with some sort of disorder, and therefore we might have a confound. For example, if you're treating pain, we know pain has an effect on neuropsychological performance, and we wanted to avoid that confound. So we focused more on non-prescribed individuals who do not necessarily have a clear-cut diagnosis that might confound our results. So let's jump into the results of our study, and what you notice in this table, this is the regional differences in FA values of the frontal subgyral regions in patients with long-term heroin use compared to healthy controls. And what you notice is that the two regions is the left frontal lobe and the right frontal lobe. So this is clearly a much more frontal with a subgyral component to it in both cases. However, what you will notice is the total voxel size is huge on the left and quite small on the right. So this clearly is telling us that the findings are much more robust on the left side of the brain. When we break down the regions, what we see is frontal lobe subgyral area with 899 voxels, the limbic cingulate gyrus, another huge voxel, sublobular extranuclear area, the medial frontal gyrus, the middle frontal gyrus, and different parts of the frontal lobe. These become smaller and smaller in terms of voxel size. On the other hand, on the right side, we're talking about simply the frontal subgyral region with some cingulate involvement. It is interesting to also look at the relationship between duration of use and the region that is most affected. And what we found is that left cingulate gyrus is associated with longer heroin abuse when you compare them to healthy controls. So the left cingulate gyrus stands out as being the most correlated with length of use. The longer people use, the more difference in white matter degradation. So obviously this is a negative change in white matter in the cingulate gyrus. So in a nutshell, so we see reductions in the fractional anisotropy in bilateral frontal subgyral regions with a specific left-sided emphasis that extends to limbic structures and to the prefrontal association cortices. The left hemisphere, as I said, is much more, the results are much more robust. And interestingly, this finding is in line with neurocognitive neuropsychological research that show that a lot of the deficits in heroin abusing populations tend to be in the left hemisphere domain that involve language, etc. It is important to always recognize when we do such research that we don't know whether this is truly the result of the heroin or there is the potential that this could be premorbid. What we need to do is more primary studies looking longitudinally at the recovery, because part of the way to tease out is that areas that recover most likely were affected by the heroin rather than having been premorbidly impacted areas. Obviously, we can also look at genetic factors. We could look at brothers or sisters. More preferably, we could look at twins of heroin users and see if they show similar. Twins would be perfect. Whether they show similar brain abnormalities, even though that they might not be using. So we would use discordant monozygotic twins for such research. However, that hasn't been done yet. Damage to tracts connecting cortical, limbic, and striatal regions seems to be really important, and they stand out in the case of heroin abuse. And specifically, it seems that the cingulum is especially impacted. So reduced frontal, reduced fractional anisotropy in, again, bilateral sub-gyral, bilateral cingulate, and bilateral limbic lobe, and left medial frontal gyrus is a summary of all of that. And we also see damage to short-range fibers in the superior longitudinal fasciculus, and that's supported by other research that shows reduced white matter in middle frontal and precentral gyri. Heroin-associated white matter damage has been hypothesized to lead to neurocognitive deficits associated with corticostriatal circuitry functions such as executive function disturbances, and what we saw in our study is that these regions are clearly implicated in the neuroanatomical changes in heroin. So next step is to actually look at whether neuropsychological meta-analyses would prove to be robust and would show us the same or the expected finding in that direction. We also have done a meta-regression that analyzed the duration of use, and that revealed that there was no significant fractional anisotropy differences between heroin abusing individuals and healthy controls who have only used for 5 years or less. However, the white matter integrity in the left cingulate, as I mentioned, and I showed you an image of that, tends to be much more significantly compromised in individuals with 5 years or more of heroin use. And so similar effects have been seen in terms of cognition, and research has demonstrated that in addition to the neurocognitive deficits that result from long-term opiate use, there is also the potential that these are associated with poorer prognosis. Individuals who use heroin for a longer period of time, who might have injured areas such as the cingulate gyrus, have more difficult time actually stopping. Moving to the question of abstinence. So what happens if somebody abstains from heroin? What we have observed when we compare individuals who have abstained for 30 days or more compared to heroin users who have just recently abstained 3 to 7 days, we discover that individuals who've had 30 or more days of abstinence tend to have very significant reductions in the left cingulate gyrus that we discovered is associated with the length of use. So it's an interesting finding, and it tells us that there is potential for recovery. However, there should be special attention given to heroin users who are put on methadone. These individuals went from heroin to nothing, and the question is, does methadone then disrupt this recovery that we see in white matter, and does that then have a functional effect? Do individuals who are put on methadone have white matter non-recovery, and is that associated with functional outcomes such as cognitive outcomes, or even more interestingly, potential for relapse if you go cold turkey? Given these findings, I think we can definitely suggest that we do more research, again, especially focusing on the difference between methadone users and non-methadone users after abstinence from heroin. We would highly recommend looking at potential process of recovery and whether that is associated with changes in cognition. With that being said, we will continue our presentations on heroin. In the next video, we will talk about recovery of changes and recovery in gray matter. Thank you for your attention.